So the FDA Immunogenicity Assessment, I literally just copied pasted the table of contents. And I encourage you not to look at any particular guidance. I am actually taping this in 2018, and I am using the latest guidance that it's available. However, these guidances are constantly updated, and they have new information. There might be new methodologies or new knowledge that can impact all of these guidances. So this is just an example, and you should look for the most current version of these guidelines if you're interested in looking at them. But what I want to just highlight is that in the table of contents for the FDA, they say, we need to understand in the risk factors what are the patient-specific factors that can affect immunogenicity. So this is from the population and the patient standpoint. Is this patient capable of mounting an immune response? Or is this patient a patient that has an immune system that's compromised? And it could be compromised because of either the disease that the patient has or the medications that a patient is receiving. So if you're looking at potential immunogenicity in a population of patients with cancer, for instance, who are taking heavy chemotherapy, you might not see the full immunogenicity ability of the drug or potential of the drug if you're looking at this population. So it is very important to understand from the patient standpoint what are the different potential risks that you have for immunogenicity. And as you can see there, it's the immunologic status and competence, as I explained. Has the patient been previously exposed or received a particular either drug or drug product? So is there any history of allergy to any of those? What is the route of administration? What's the dose? What's the frequency of the administration? All of that has to do with the response of the immune system to the drug. There might be some cases with genetic predisposition to a particular immune response. And what is the status of the immune tolerance to endogenous protein? So in part 2 of this series, I did explain a bit about the importance of having a drug that could be an endogenous protein. And if you have an immunogenic response to an endogenous protein, this can carry consequences in terms of safety of the patient. I believe I also mentioned a little bit on part 1, but basically you can find more explanations on this specific on part 1 and part 2. But for part 3, what we need to focus on is the fact that if we are looking at a drug that's an endogenous protein and it is immunogenic, it can have consequences on that patient that go beyond just the drug administration. You can always discontinue the drug if there's a particular problem with immunogenicity. But the problem is if your drug is an endogenous protein and now the patient has mounted an immune response to the endogenous protein. So you're given insulin, the patient reacts with immune response to that insulin that you're giving the patient. You can stop the treatment, but maybe the immune response is now reacting to the patient's own insulin. This can happen with many different kinds of proteins. So it is important to understand what is the status of that immune tolerance to the endogenous protein. And you should read the package insert of the drugs you're using to understand all of these aspects that are important in immunogenicity. And then there are product-specific factors. So you have patient and product. So from the product standpoint, the type of product, formulation, chemical characteristics, biologic characteristics, physical characteristics, all of them are super important because they can all trigger a potential immune response in a given patient. And the most important thing is that ultimately what we need to understand is if the immunogenicity of that particular drug is having any consequences on efficacy or consequences on safety. When you look at the European Medicines Agency guidelines, again, this is the one from 18 May 2017. We're now in 2018, and these are the latest ones that are available, but these are documents that can get updated at any time. But basically what you see is, again, there's the highlight of understanding what are the risk factors that can predispose an immune response, and it's from the patient standpoint and the product standpoint. So it's different in order of where the FDA requires their package to be, but they both talk about the same thing. Understand the risk factors from the patient perspective and from the product. And understand were there any consequences on efficacy and were there any consequences on safety. And this is talking about the whole immunogenicity. So in terms of the consequences of the product, is important to understand if they also affect the pharmacokinetics, not only safety and efficacy. This is also included in the FDA recommendation. So bottom line, the two agencies have a lot of things in common, and they both recommend understanding the risk factors from patient and product, and also understand the consequences that the immunogenicity are having on efficacy, safety, and PK. So from the FDA assessment, when we're talking about safety, it is very clear that the agency, when we talk about anaphylaxis, which is a serious adverse event, the agency relies on several different diagnostic criteria, but they strongly recommend using the Samson criteria, and I will be explaining a bit more about what the Samson criteria is. As a clinician, if you have a patient and you're going to dose that patient, understand the potential immunogenicity reactions when you read the package inserts, understand what would be expected, and make sure that you follow that patient. You keep the patient in observation depending on the drug that you're using, if there's some anaphylaxis seen with the drug, and make sure that you can capture any potential signs and symptoms. For the drug approval, the agency, the FDA, is expecting to see the Samson criteria applied, which I will explain later. But from an individual standpoint, you can actually look at any kind of potential adverse events of interest to analyze this potential anaphylaxis to the drug. A very important thing is anti-drug antibody alone is not necessarily predictive of an immunogenic response, especially anaphylaxis or other hypersensitivity reactions. So this is critical. Having a patient who has no anti-drug antibodies does not tell you that there was not an immunogenic response. We need to make sure we look at the whole manifestation of immunogenicity with or without anti-drug antibody present. In the European Medicine Agency, what they want to see is an integrated analysis of the clinical significance of the immunogenicity. So basically, when a sponsor is presenting a program, there are many different protocols, trials, and results, and what the EMA agency is expecting is seeing a standalone document that has integrated analysis of the immunogenicity but clinical significance. So what they focus on is not just was there immunogenicity or not, but what was the clinical significance if there was any immunogenicity. So that's the important thing, is clinical significance. And in this integrated document, they want to see effects on pharmacokinetic, pharmacodynamic, clinical efficacy, and safety. So not just saying this is the immunogenicity response and that's it, but was there an effect on any of these particular parameters that was of clinical significance? So there are key elements that differentiate the two approaches, but there's more commonalities between them than differences. The specific differences is, as I mentioned, the European agency is for now requesting or expecting an integrated summary of immunogenicity as a standalone document. Even though the FDA has not yet, as of the guidelines in 2017, requested it or expected it, all of the elements in the integrated summary of immunogenicity that the EMA is expecting are also expected by FDA. They could be in different documents, they could be in summaries of efficacy and then have a section on how the immunogenicity was impacted, summary of safety, and then have a section on the immunogenicity. And they definitely are singling out the Samson criteria used for analysis or assessment of anaphylaxis, which the European agencies basically says, remember to make sure you consider the potential anaphylactic reactions, but they're not telling us to use the Samson criteria.